So, George, thank you kindly for hosting me here at your lab on this very blustery day here in Boston. Before we get into genomics, I'd love to talk briefly about your background. You've been here at Harvard almost continuously since the late 70s, am I right?
That's right. I was a graduate student from 1977 to 84 and then professor from 86 to present.
And I looked at the list of startup companies whose roots trace either directly or partially to your lab, and it's immense. It looks like it's pushing, almost 100?
Well, so I've been involved in easily 80 companies. I've co-founded on the order of 22, depending on how you count the most recent ones, which aren't fully founded yet. But usually it was less than one a year for a while, then it was two, and this year it's already 11.
Wow.
Yeah, and most of them recently have been postdoctoral fellows leaving the lab, often with other people in tow, starting their own companies. So it used to be that I'd be doing it with some other professor, some other university, with a VC involved. Now it's much more homegrown than ever before.
Wow. And if this new homegrown methodology has spawned 11 companies this year out of 22 over a span of decades, it certainly sounds like a productive approach. Anyway, as you know, my introduction to this interview will include a pretty robust overview of the topics you and I now plan to cover, which are genetic sequencing, genetic editing, DNA synthesis, and DNA assembly. It'll also include good starting point definitions of all those things. So that background has already been transmitted, which means you and I can dive right in. So let's start with sequencing. When I think of sequencing, I immediately and naturally think of the Human Genome Project because of its renown, its size, and its audacity. You were, of course, deeply involved in it, literally going back several years before its beginning. What's your big picture take on the Human Genome Project now, 15 years after it concluded?
So I personally don't think it was audacious enough. I think it was misdirected, and I'm disappointed. I felt from the very beginning, 1984, when we first proposed it, the goal should be to reduce the price, ideally to something that was too cheap to meter, like $1,000.
So to work on the technology and make it much cheaper as a first step.
Yes. And then start sequencing genomes. Maybe do a little test along the way, but not aim to spend $3 billion, which was our original goal. A number we kind of pulled out of the air, $1 a base, for one genome. A clinical genome today would be essentially two genomes, your mother's contribution, your father's contribution to you. And so the thing that we did for $3 billion was one of those, which would not be useful clinically. So we didn't actually produce a useful kind of genome. So that meant that we had to develop a whole new set of technologies afterwards. That I consider the audacious project, which was making a $1,000 genome that was diploid, meaning both your mother and your father's contribution.
And this perspective of yours has amazed me ever since you and I first discussed it on the phone a few weeks ago, because I'd always imagined that the Human Genome Project was a 13-year period in which there was breakthrough after radical breakthrough, and things just got cheaper and faster. But in reality, it sounds like it was quite conservative in terms of the technologies that were developed and deployed.
I considered it crazy conservative. See, the cheaper and faster are two very different things. You can always make something faster by just buying more machines.
In other words, by spending money faster.
Yeah, and that's almost exactly what we did. There were some minor improvements, but to tell you how minor they were, essentially none of the improvements during the Genome Project persisted.
So then technologies were created during that 13-year period, but they were generally too timid or not transformative enough to last until today?
So there were a few things before the Genome Project that persisted, like shotgun sequencing. We still do that. But all the innovations on electrophoresis, which I consider quite incremental, like changing from slab gels to capillaries, those are mostly gone in what's next-gen sequencing. Next-gen sequencing was a complete redo, which was based on microscopy.
And just a quick definition for listeners, next-gen sequencing is sort of a blanket term covering a raft of innovations that emerged after the Human Genome Project. So in light of all that, what do you think of the $3 billion investment in the Human Genome Project?
So basically that was thrown away because neither was the genome of the clinical grade, nor were any of the technologies preserved.
And to put the power of post-Genome Project technology, in other words, next-gen technology, Into perspective, the project itself spent $3 billion over 13 years sequencing a highly imperfect genome, and it ended in 2003. How does that compare to the cost and time requirements of sequencing a much more robust human genome today?
Right. So probably the lowest price right now is $600. The timing, it depends on how you count the DNA prep and various other things, but it's short. It's in days now rather than in years.
So that's well over a 3 million X improvement in price over the 15 years since the Human Genome Project ended, a time period which is almost equal to the time span of the project itself, interestingly. So what do you think would have happened if the focus had been on the technology first? Do you think we would have gotten to the endpoint of the Genome Project much more quickly?
Oh, yeah.
Much more quickly.
Yeah. And we would have had $3 billion to generate genomes. So we still haven't spent $3 billion on clinical grade genomes.
In the time since the end of the Genome Project.
In the time since then, because we kind of burnt it up on the first one.
So why did it play out that way? It sounds like you, for one, clearly wanted to develop the technology first.
So I didn't have much of a vote. I mean, I was the youngest person in the room in 1984. And I was probably the youngest person with a grant when the grant started getting handed out in 1987. And I think the reason wasn't evil or anything. It was just that there were very few engineers at the time in biology. And the few engineers had no clue about molecular biology. So there might be engineers that were working on artificial joints or something, or maybe engineers that could build a better patch clamp for neurobiology, but very, very few in molecular biology.
And so when the Genome Project ended, there was what you recently described to me in a prior conversation, a technological overhang. A lot of developments which had been partially made under the radar in skunkworks burst into the light and started getting fully fleshed out. And that's why we've had this crazy acceleration since the end of the Genome Project. So where does it go from here? Do you see another 3 to 5 million X improvement over the next 15 years? Or are we going to run into the limits of physics?
Well, ultimately, there will be some limits of physics, and you can get some idea of that from how quickly and cheaply you can transfer data from one form to another, like transferring it from a Blu-ray to a RAM in your computer.
In other words, we should one day be able to transfer a genome's worth of DNA into digital storage as quickly as we can transfer from one digital medium to another. So what does that say about the eventual cost of sequencing a genome?
So transferring a genome equivalent, which is less than a Blu-ray disc, into your computer is a penny. It's probably the electricity and plus the amortization of your laptop. That's where we're headed, is my guess.
So we're really in a headlong rush to the dollar genome, then to the penny genome, and maybe even to the sub-penny genome.
And as far as the consumers go, long before we get there, the consumers will be seeing $0
Right.
and maybe even being paid to get their genome sequenced.
Because that data will be valuable enough to other entities in the system to merit that?
It's terribly valuable already. Right now, we've already gone past the point where all of us should be paid to have our genome sequenced because the system could make on the order of $1 million every time we save one child from a serious Mendelian disease. That $1 million should be then spread out to all the people who participated, including all the people who didn't get any bad news, right?
In other words, if detecting a disease early can nip it in the bud and save the healthcare system $1 million, if just one person in 1,000 has a detectable disease, even with today's cost structure, we're already ahead of the game if we sequence everybody. And then certain diseases can be avoided entirely if two people who both carry a dangerous recessive trait figure that out before they create a child.
So if you get the carrier status, you can avoid other carriers of the same thing. And to some extent, at that point, you and all the people who didn't get carrier status should be equally compensated.
So all this takes us well beyond the definite article in the Human Genome Project, when it was really a human genome was being sequenced.
Not even.
Not even. It was half of one, right? So let's talk about what happens when we go from a human genome to hundreds of thousands and eventually millions of complete genomes in a database. There are now several projects underway to create giant databases, one of which you initiated, the Personal Genome Project. What do you imagine we'll be able to achieve with the vastness and the depth and the richness of the data that we'll have, let's say in 5-10 years, with millions of genomes sequenced and also ideally matched to phenotype and health records?
Well, the critical thing is that coupling with phenotypic information, the health records and environmental components, big environmental component of the microorganisms and viruses you're exposed to, and how your immune system responds to them. If you have all that, almost everything we die of is genetic. Now, some of it is both genetic and generic. We're all going to die of age-related diseases, at least the lucky set of us that live in industrialized nations and escape infectious diseases. But we will get insights into all of human biology. And many of the solutions won't depend on us necessarily finding the cause and reversing that cause. Some of them will have completely synthetic approaches. In fact, most of pharmaceuticals really are not reversing some natural process. Literally, it's coming up with something kind of out of left field that's used at a pharmacological level, not at a physiological level. Synthetic biology is a better paradigm than natural population genetics or natural human genetics.
But natural population genetics will give us major insights because we'll start being able to do much, much bigger correlation studies. Of course, we've already correlated thousands of genes with thousands of conditions, but those correlations are imperfectly understood because we don't yet have millions and millions of genomes, and we certainly don't have them matched to phenotypes and health records. But when we do start getting into the millions of genomes sequenced, is there a point at which our knowledge becomes so statistically significant that there's very little value to adding still more genomes to the database? I mean, is there a threshold beyond which we kind of have enough genomes to understand human genomics? Or if it's possible to sequence the next, then the next, then the next person, it'll always be scientifically beneficial to do that?
We have 7.5 billion people on the planet. I don't think that's enough. I think it's a good argument for having more babies so we get more insight into human biology. Now, that's a little facetious, but the point is, it's not just about correlation, it's about cause and effect. And we can learn a lot from one person. So in a certain sense, if you have one person, you can test cause and effect. You can follow up on anything that looks bizarre about that person, about their genetics. And we're getting better and better at determining cause and effect. And that may, in the end, be more powerful than correlation from billions of people. The thing that's interesting about having 10 billion people or 20 billion people as your resource is that you can find really rare things which are N of 1. They're really interesting N of 1 cases where they've had an unusual environment or they've had some weird combination of alleles and it makes them unique. That's what I think is the advantage of having more people, not necessarily moving the number of times I've observed a particular allele from 1,000 to 10,000. That points to diminishing returns.
And if you find an N of 1, a unique one-of-a-kind case we might discover upon sequencing, let's say, the 7 billionth person, the scientific value wouldn't be in discovering yet another rare disease, right? I mean, clearly you'd want to cure that person if you did find a disease in them. But from a public health standpoint, that disease would be far too rare to move the needle, right?
I'm less intrigued by the N of 1 being a rare disease we're going to cure. I'm more intrigued by the N of 1 being a rare protective allele that's going to save us all. For example, you can find incredibly rare people that are resistant to HIV. Some of them are resistant because they don't have receptors on their T cells, and some are resistant because they got a fluky antibody. And my lab participated in some of these studies that studied these exceptional people that would escape HIV. Those antibodies could be powerful protection for the rest of the planet. Or you get people that live to 122 years old. Maybe they have something that all of us could share. Maybe there's a dozen of them that each have a different thing. And each of those N of 1 gives us one more tool in our effort to reverse aging so that we have essentially the body and mind of a 22-year-old, but the experience of a 130-year-old, right?
So that's why the hypothetical 20 billionth person could be the source of a great discovery. Now, when I first started thinking about personal genomes, maybe 10 years ago when 23andMe started getting a lot of press, and I started realizing, wow, someday we're going to have $1,000 genome and then a $100 genome. Back then, I assumed there'd be some kind of endpoint beyond which cheaper and cheaper sequencing wouldn't be very useful, because what do you do once we're all sequenced? But of course, it turns out that there's lots of useful things that go far beyond personal genomes, which can only be enabled by ever cheaper technology. So I'd love to discuss something you've speculated about, maybe in the 5 to 10 year future, which is genetic monitoring, a system that could let you determine if there are unfriendly pathogens in a room as you enter it or even before you enter it. What kind of device and horsepower would we need to pull that off?
My gut is that it's going to be some electronic sequencing. So either nanopore or some other single molecule electronic, which has very low reagent costs, very small fabrication costs, possibly portable, possibly disposable. And you could like scatter them around the room. You could scatter them around your body. They could be that inexpensive. And then you've got social network that's exchanging information about where all the pathogens are in various rooms in various parts of the world. And it would really revolutionize epidemiology because it changed it from being something where you do a 10-year study to something where you do a 10-second study. And you know whether you're sending your kid to daycare with H3N2, some deadly flu virus, or norovirus or Ebola, rather than some minor rhinovirus or coronavirus. It's just going to cause a little sniffles in everybody in the class.
So this would be enabled by extremely fast, extremely small, extremely cheap sequencers, which would basically inhale pathogens out of the air and identify them. How many years off would that level of robustness be? Are we talking decades, or is this in the more intermediate future?
I think almost nothing we're talking is decades. We're already at the point with the nanopore sequencing, you can put in samples and start getting answers out in 25 minutes. The DNA preps are really easy, so it's increasingly integrated with the device. And so the step from that, from sampling something from the air, tends to be pretty clean. So you would get pollen, you get some viruses, a few bacteria. It's pretty clean. But anyway, all that would go into a simple deproteinizing and into the nanopore. I think we're talking about years. And the software is not hard either.
So single-digit years.
Yeah.
Interesting. And if you can already do this kind of test in 25 minutes, the path to 10 seconds seems daunting, but that's only, 2 orders of magnitude in a field that routinely advances 5 or 6 orders of magnitude in a decade or two.
Yeah. Speed you can gain by parallelization. Really, the process of stripping proteins from a virus is pretty fast. I mean, it really can be instantaneous. And then threads into the nanopores instantaneously. That is very close.
And device size could be something that just sits on your smartphone or hangs around your neck.
The device size already that thousands of people are using is 70 grams. It's the size of your cell phone.
And obviously, that's going to shrink. On a kind of related note, you know Danny Hillis, right?
Yeah.
Are you familiar with this app he created called Dark Sky? It's a cool weather app which leverages the fact that most smartphones actually have barometers built into them. So the millions of phones that are running Dark Sky constantly report back barometric readings, and those readings are tagged with GPS coordinates, which lets the system give you a reasonably accurate minute-by-minute forecast for your tiny microregion, like maybe a 100-yard circle. I don't know. In other words, it also leverages detectors that millions of people in the network are carrying.
Well, there's a similar thing for airplane navigation and, of course, for car navigation, Waze.
Exactly. So your detector network would be like Waze or a Dark Sky for disease, and it would get more and more powerful and accurate as more and more people adopt it. That's amazing. And what do you think the 2.0 version of this might do, as if that's not already amazing enough?
Well, I think one thing is preventing us from spreading known pathogens. The other is discovering the next pathogen before it's an epidemic. You can notice it's going through birds or pigs or something like that. There's detecting your immune response to it. So Steve Elledge and I worked on methods for checking your autoantigens, and that could be something that's close to real time. And so you could start connecting. What do I get exposed to that causes me to become autoreactive? Because I think a huge number of diseases that we have that are not well explained right now are actually our interaction with our environment and our immune system. Probably a lot of things that cause chronic fatigue or arthritis, gastrointestinal problems, diabetes, and so forth are complications where part of it is genetic, but a huge part of it is immune response to the environment. And so I think that we could get much better at that.
By doing some sort of monitoring inside the body?
Well, actually, the way that Steve and I did it is you take a lance, just like diabetes test, and you need much less than a drop of blood. And you can do this whole assay for every possible virus, every possible human antigen. But yeah, internally would be even better. In fact, it'd be really great to embed a lot of sensors because it's all getting sub-millimeter circuitry. There's no reason why you couldn't make something that's very non-invasive but is monitoring 24/7.
So now that we've talked about sequencing, let's get to part 2 of our discussion, editing. Editing will continue to matter hugely until the somewhat distant day when we can make arbitrarily long, error-free DNA strands, because editing lets you start with an approximation of the endpoint you're targeting, assuming that approximation's already largely built into the genome of a virus or bacteria or an animal or whatever. Then you just have to make relatively small changes to reach your goal. CRISPR's gotten a huge amount of press and adulation as an editing tool, but it certainly isn't the first genetic editing tool. Could we talk a little bit about the heritage that preceded CRISPR and why CRISPR is a significant breakthrough, but also why it's not the last breakthrough and why there's a lot of headroom for improvement from here?
CRISPR looks to me to be a momentary pause rather than a gigantic breakthrough. And I can say that because it's partly my baby, shared experience with many other. And the earliest efforts in mammalian cells, at least, were groups like Mario Capecchi and Oliver Smithies, who would introduce double-stranded break linears into the, say, mouse cells. And they would, at some low frequency, like 1 in 1,000, 1 in a million, get incorporated into the right place. And then you'd have to screen through those, and then you could construct a mouse that was exactly what you wanted. And in a way, that 1980s technology, they got the Nobel Prize, was better than CRISPR in certain ways, in that a mouse was exactly what you wanted. What most people call CRISPR editing is knocking things out. It's not precise editing, it's kind of bashing. Along with and after the Smithies and Capecchi, then you had a series of enzymes that would help you soften up the genome a little bit. So moving it from 1 in 1,000, 1 in a million to more like 1 in 100. And those were meganucleases, which Bernard Dijon, one of my advisors, discovered in the 70s. Then zinc fingers and zinc finger nucleases. And then taus and tau nucleases. So those all would cut, but there was a race between fixing the cut with what you wanted, perfect edit, and just making a mess. And then there were another set that were much better at doing what you want without making a mess. In other words, instead of making a double-strand break, they would swap in the DNA. There would be a more dance-like cooperation between the donor DNA and the recipient.
So it was more of a pasting operation than a cut and insert operation.
Exactly.
Let's talk about what makes CRISPR exceptional versus previous editing methods, both quantitatively in terms of price, performance, and speed, and also qualitatively in terms of things that it's good at that its predecessors weren't good at.
The two main things would be price and efficiency. Almost nobody makes their own CRISPR. They purchase the enzyme in the form of a gene, and they purchase the RNA that goes with the enzyme custom. And so you pay some custom price, which varies depending on how smart you are at getting the right vendor. But it's in the order of hundreds of dollars. And maybe the talons are $1,000 or something like that.
And to clarify for those who don't know, talon is a fairly new approach to gene editing whose popularity peaked right before CRISPR. And it sounds like CRISPR is cheaper, but not radically cheaper than talons. So clearly then CRISPR's advantages go far beyond cost.
So then efficiency is the other thing. There was a nice study early on. The same sites were targeted by talons and CRISPR. And on average, it was about a factor of 4. But our paper was the first one where we compared talons and CRISPR on the same site, and we had a factor of 20. But that was one anecdote. This paper did 9 different sites, and it was an average of 4.
So the efficiency went up by somewhere between 4 to 20x, which is substantial. And I sense that efficiency has a very precise definition here. Does it refer to yield?
Once you get it into the cell, what's the probability that cell gets altered?
In the way that you want.
In the way you want.
Okay, so CRISPR improves efficiency by 4x or more. What odds of a successful alteration does that map to in raw percentage terms?
It depends on the tissue. It depends on the organism, the particular way you do it. It can get up into the 90% range, but some tissues is still in the 1% range. So what was that factor of 4 was going from, say, 10% on average to 40% on average.
So 4x is clearly a very respectable improvement, but you've seen much bigger improvements on other fronts.
You know, we get excited about that factor of 4, but then there's that factor of 3 million, which is the reduction in cost of sequencing.
So there is substantial room for improvement, not only in efficiency, but in the elegance of the process, because you've been quoted saying that in some ways CRISPR is almost more like vandalism than true editing. And a moment ago, you compared it to bashing. How does CRISPR fall short of an ideal system that would do true editing?
Well, you can get it to put in what you want. You put the donor DNA, and then you cut the recipient, and then you hope the donor DNA diffuses to the site where the recipient's been cut to act as a bandage to repair it. But the host can't wait. And the host cell is saying, hey, there's a double-strand break. I'm freaking out. Let's fix it. And they fix it by a process called non-homologous enjoining, which typically removes a base or adds a base or 2 or 3 or 100. And you get some random thing. The previous set of gene therapies were additive. You're genetically missing a gene, your gene therapy adds it back. So you needed to be able to subtract. And then the third thing you want to do is do whatever you want. You want to be able to do something very precise. So let's say sickle cell has a particular base pair change, you want to change it back. And that's neither addition nor subtraction. It's precise editing. And if we had precise editing that was cheap and effective, we'd do everything that way. If you edit a document, you don't want to say, oh, I can only remove some random number of letters from a word, right? You can say, well, that word's wrong. Let's just change it to some other word, or let's just delete a few letters. You want to change it to exactly what you want.
So in a sense, for all its great improvements, CRISPR is still kind of like being able to swap out a paragraph. Like I can take out a paragraph and glue in a more perfect paragraph, but I can't dive into a paragraph that only has 2 wrong letters and precisely change those 2 letters.
It's just inefficient. So many of the projects that my lab wants to do, and many other labs, is not change one gene, you want to change many of them. And it's so inefficient at doing one that it's hard to even imagine doing multiple ones.
And the reason is, just to make sure I'm understanding this correctly, if you've got a 40% chance of making any one particular change successfully, your odds of making 2 changes is 40% of 40%, or just 16%. And your odds of making 3 changes is what, about 6.5%. So if you want to make 50 changes, your odds of pulling that off are 40% to the 50th power, which is essentially 0.
Yeah.
So to do hundreds of changes, you really need to push your efficiency up to like 99% or something. Plus, you want to become surgical as opposed to additive and subtractive, which seems like a tall order. But given that we've already had 10 different types of editing since the 80s, we'll probably have 10 more in the next couple of decades. And at some point, we'll get there, right?
And it'll probably be fast, too. I mean, talons lasted for about 3 years before they were replaced by something else. It's already been 5 years since our first publication of CRISPR as a technology in 2013. We're overdue.
And I'll add that CRISPR almost seemed to come out of nowhere, and not just to outsiders, because I also know lots of people who are very deep in the field who just didn't see it coming. Now, for you as a super deep insider, how clear or opaque is the pipeline of future editing technologies today?
Some of these things look faster to people that are far away from it than people close to it. For example, fluorescent next-gen sequencing looks like it came out of nowhere. It came very quickly. But I watched it slowly develop from our first paper in 1999. But for most people, it's suddenly popped up in 2005, okay? But that's still pretty fast. And with CRISPR, to me, it was on a continuum with all these other methods, the Smithies and Campeche and the meganucleases and so forth. And so it looked slow to me. It was another factor of 4. It already had several factors of 4 from Smithies and Campeche stuff, which was 1 in 10,000.
It's interesting. I had an interview with Rodney Brooks a couple weeks ago, and when we were talking about AI, he made a similar point, which is that while neural networks and backpropagation seemed like sudden and radical developments on the outside, the field had actually been kind of pregnant with them for years and years and years. So when they did finally happen, it did seem like a completely discontinuous change to us non-AI people, but it was actually nothing of the sort. Anyway, moving on to the practical applications of editing, I'd like to talk about xenotransplantation, partly because it's such a cool word and I feel smart whenever I say it, but mainly because it has extraordinary potential for humanity and it's being enabled by these advances in editing. So let's talk about what xenotransplantation is, why progress in it came to a grinding halt about 15 years ago, and how these breakthroughs in editing that we're getting now might get it back on the fast track.
Right. So it was a great idea about 20 years ago that if you look at pig organs, they're all about the right size and shape to replace all the organs we get currently by going between people. For a while, it seemed like the problem was finding a match. But it's mainly there just aren't enough people that are dying under circumstances where they can donate organ. Usually they've got multiple organ failure or they're really old and they're not good donors.
And millions of people die awaiting a transplant.
Exactly. I mean, there are millions of people that could benefit from transplants. Some of them aren't even considered seriously right now because there's such a shortage. They're going to say, well, you're in such a long queue, we're not even going to call you part of the queue, right? You have a kind of disease which will just recur if we give you an organ. And 20 years ago, the idea of the pig was there. And I think it was quickly appreciated. When you transplant, you get a very acute reaction, which goes beyond the kind of transplant reaction you get between humans.
And by acute reaction, you mean the body rejects it?
Rejects it very fast, in hours, not in weeks. And that's because pigs have different sugar on their surface of the cells. And then they figured out a way to fix that. Well, that's one gene. And that did get rid of the really fast reaction, but you still had all kinds of slower reactions. And there were clearly multiple things wrong. And the other thing that killed it 20 years ago was that it also slowly dawned on people all the pig cells in every pig in the world were producing viruses.
The cells themselves were producing viruses.
And it wasn't because the pigs were infected. It was built into the pig genome. So it was a natural property of being a pig is that you make viruses. And it's not just pigs. It's many mammals do this. And so this is a worst case scenario. Put an organ from a pig into a human, you save the human's life temporarily, but the recipient is immune suppressed because that's the way you do graft transplantation.
You put them on drugs to knock their immune system down so they won't reject the organ.
But now they also won't reject the viruses.
So now there's a swarm of pig viruses in our patient
that are growing like gangbusters because they're immune compromised. And that means they can start evolving.
The viruses can start evolving?
The viruses start evolving in that patient, and then they could infect healthcare workers and family members. And you would not want to be the provider of that technology.
Nor would you want to be the person changing those sheets or serving that poor, ailing patient breakfast.
So that caused a slowdown.
And it was $1 billion effort in the 90s, wasn't it?
Novartis and many other companies were investing more than $1 billion, and it was worth it. It was considered a good effort. And then when we invented CRISPR, we suddenly got, within a few weeks, 3 different pioneers, people that really had lived through this era and had dreamt the dream for a long time. And they each came to us independently and said, hey, we'd love to have you use your CRISPR because we know there's not one gene. There's lots of genes we got to change, maybe dozens. Do you think CRISPR is up to it?
And they knew roughly where on the pig genome these viruses were lurking?
The viruses and the immune functions and the blood clotting and so forth. Either they knew or they knew somebody knew. And so they recruited us and we started looking at the problem and we said, yeah, this looks like a good choice for CRISPR. Although it was still pretty challenging. At the time, we and others were changing 1 or 2 genes at a time with CRISPR, and we didn't even know how many viruses there were. So the first thing we did was sequence a bunch of pig genomes to figure out how many viruses there were. And in the pig cells that we were going to test this on, there were 62 viruses. So we had to make 62 at once. And, we just went through the calculation of how inefficient CRISPR is. But we said, well, we got to try it anyway. It seems like it's going to be the product of a bunch of inefficient steps, 0.4 to the 62 power. Doesn't seem very hopeful, but we're definitely going to fail if we don't try. And it turned out it was much easier than we expected, embarrassingly easy. So 14 days in the incubator and almost no human effort, and then a little bit of PCR screening, all 62 of the viruses were now damaged in the way we wanted them to be damaged in the polymerase gene. But that was just the first part. Those were done in abnormal cells. People have a tendency to do CRISPR on cells that replicate well, that are abnormal. And so we had to redo it on normal cells so we could put it into a pig and develop baby pigs. And that worked.
So there are now baby pigs.
The first pigs in the world that have no functional endogenous retroviruses.
When were they born?
They were born in early 2017.
And where are they?
We're developing them separately in America and China because it's actually hard to move animals from one country to another.
And you're in both countries because you started a company with a Chinese co-founder, right? What's the startup's name again?
eGenesis is a company that we founded after we started this project, along with Luhan Yang, who was a graduate student and a postdoc on my lab and one of the co-inventors of CRISPR as a technology. She is now chief scientific officer there. And they're not only piglets anymore, they're grown, sexually mature animals having their own babies. And so we think that particular step is not damaging to any step in the life of the pig. In parallel, made a lot of changes that improve the immune quality and compatibility with humans and clotting, a number of things that make them more compatible. And now we're putting together the final pig that will have all of these things in one pig, and then those will be used for testing in non-human primates.
And if things keep moving along as they currently are, how long do you think it'll be before the first pig organ transplants will be made into humans who desperately need them?
We need to get the non-human primate trials done, but that takes about 6 months. And then it could be a year and a half, 2 years before we're doing human clinical trials. People can get fixed. Just because they're clinical trials doesn't mean you can't be fixing a lot of people.
So human trials could be just a few years off. And if this works, I mean, tell me if I'm being overly optimistic here, but it sounds like if this works, the organ shortage is over.
Yeah, it sounds crazy, but yeah, that's what it means. And it's even better than that. It sounds funny to even say that, but it's better in the sense that because these organs are engineered to be compatible, for the first time in history, you don't have to find a match. You're essentially engineering a universal organ. So you could have stockpiles, you could have them ready, and they're fresher too, because one of the problems with transplants is you'll get an organ in as fast as you possibly can from some motorcycle victim or something. And it was in pretty good shape when it's taken out of the victim, although not perfect because you can't take it out until the victim is declared dead. And then there's a delay of shipment. And then the surgeon has to decide, well, it's a little gray. It's not so red. Should I use it? And so then you take a chance of hurting the patient that you're transplanting it into. This would eliminate that. But it's even better than that in that because they're universal and they're engineered, you can now make organs that are resistant to pathogens, resistant to senescence, resistant to cancer, and compatible with cryopreservation. So you could even have these things frozen, ready to use immediately. So you don't have to have a living pig in the surgical ward.
So if you're in the unfortunate situation of desperately needing a kidney transplant, you not only get off dialysis and get a completely new lease on life, but now you're never going to die of kidney cancer. I mean, you could die of another cancer, but this organ at least is pretty safe.
It opens up that door. This is not a promise. We've made animals that are resistant to certain pathogens. We've made animals that are resistant to senescence and cancer. It would be very hard to engineer a human to have these enhancements. But if you're going to transplant the organ anyway, you might as well give it the best organ you can.
And this whole development is very well timed because the organ shortage is probably about to get a lot worse, albeit for a very positive reason, which is that self-driving cars are expected to radically reduce the number of traffic deaths, which are our main source of organs over the coming decade.
And this already happened. We reduced the number of accidents already with seatbelts and airbags.
Now, speaking of pig transplants, you're vegan, right?
I am.
And does the notion of raising pigs for the purpose of accessing their organs sit awkwardly with your reasons for becoming vegan?
Well, I'm vegan for a variety of reasons. The main one is a health reason. I think many people could benefit from a low cholesterol diet. I benefited more than I did from statins. That's number one. Now, there's humanitarian components too, but I consider that one pig saving 10 people makes a whole lot more sense than one pig producing bacon and pork chops that are going to hurt 10 people. But all that said, the ultra-vegan in me would say, this is a stopgap procedure that is going to save millions of lives. But let's also start getting organs from human tissue culture, meaning lab-produced organs, which we are actively working on, and we've been working on since before the pig. And if those arrive in time, fine. If they don't, then for years we'll be depending on pigs, and maybe forever. But the point is that as a humanitarian, I think that we need to try everything we can to prevent human suffering and minimize the pig suffering. We want to get as many organs as we can per pig and arrange it so the pigs have no pain, so that they have a happy life.
Now, there's another even more ambitious editing project, which is the bacteria which is resistant to viruses.
Ah, yes.
So let's talk about that.
That's the trick here is a very simple idea, which is you change the genetic code. That is to say, how you go from DNA to RNA to proteins, which is a triplet code. So there's 4 bases, ACGT, and they can be combined in 64 ways to make triplets.
And a quick review for those whose biology is rusty, the total number of 3-character strings that DNA can make using its 4-letter alphabet is 4 to the 3rd power, or 64. And each of those 64 combinations represents one of the 20 amino acids that life runs on, or the stop command, so a total of 21, which leaves plenty of room for redundancy. Because, for instance, 4 of the 64 combinations code for the amino acid glycine. So you don't necessarily need 4. 2 code for glutamine. 3 combinations indicate a stop command, etc.
Right, exactly. And so you can move the code around in a way that doesn't hurt the cell. So you can take TAG and turn it into a TAA, and it still does the stop codon function. You've freed up
TAG entirely because you have 2 other codons that can say stop. And so as for TAG,
you can eliminate its use from the whole genome. And that way, when the viruses come in, they don't know what to make of that. And 4 out of 6 viruses are completely baffled. They can't replicate.
They're in an environment in which this triplet is just not used at all. And that throws their metabolism off so much that
they cannot replicate. Even if you've put in viruses at very high titers, like 10 to the 12th, trillion viruses, you get nothing out. So they're really, really resistant to these viruses, but only 4 out of 6. And we made that genome, we tested it, and it's amazing.
This is E. coli?
This is E. coli, which is an industrial microorganism which has a virus problem. And that required 321 changes out of 4 million base pairs. We're redoing that. So instead of 4 out of 6, it'll be all viruses. And same trick can be used in every organism that has a virus problem.
And how far along are you with this E. coli project?
We're mostly done with it. That was done almost entirely without CRISPR. And we're now scaling up to do that in other organisms. So industrial microorganisms, dairy microbes, agricultural plants and animals, and human cells for transplantation and pharmaceutical manufacturing.
So you've made this strain of E. coli essentially bulletproof. Where do you take it from here?
We're starting to do it in human cells. Mammalian cells, like human cells, are used for manufacturing a lot of pharmaceuticals, like human proteins that have to be properly coated with carbohydrates. It's also used for manufacturing vaccines and for transplantation. And each of those has either a risk of getting viral contamination, which would be really bad for a therapeutic, or if it's being put into a human, it's getting exposed to viruses all the time. So there are reasons to make human cells that are multivirus resistant.
So let's move on to part 3 of our agenda, which is synthesis. The synthesis of oligos. How has that accelerated, improved, and cheapened since the Human Genome Project? I understand by some metrics, synthesis has gone even faster than sequencing.
So to give some points on it, in my lab when I was a student, we ordered a piece of DNA that was 10 long, 10 ACGs and Ts in a row, for almost $4,000. Today, for $4,000, we can get essentially a human genome's worth of oligonucleotides. So we can get billions of base pairs for $4,000. So it's gone from 10 to 3 billion, more like 300 million fold rather than 3 million fold.
Wow. And when oligos are made, it's in sequences that are extremely short compared to any genome, certainly compared to the 3 billion letters in a human genome, right?
200 is kind of a limit, but then you can assemble those into bigger pieces.
So why do you have to start with these relatively teeny building blocks? Why do they top out around 200 letters?
As you go from 10 to 100, you're accumulating errors. So the efficiency per cycle might be 99.5, and then to the 100th power is you start getting a lot of errors. But then as you get to 200, it starts to get even worse and worse, as if it's kind of folding up on itself and things are becoming less efficient.
Now, this might be a bit of a false separation, but I think of those short oligo building blocks as being synthesis, and then the assembly of lots and lots of them into a single long strand as being a second thing, as being assembly. Now, obviously, it's the combination of these two steps that result in long strands of DNA, but it seems like different entities do the mass production of short oligos versus the assembly of long strands. So it's kind of two different steps, two different expertises. Is that right?
There are some companies that specialize in the short oligos, and most of the really big constructs, things that are in the million base, multi-million base range, are being done by academic labs.
And do you expect the price curve for oligos to continue to drop at this blistering rate for years to come?
I do, but I can't guarantee it. I think both the reading and writing curves have quite a bit left, many factors of 10.
Now, you said that the very skilled artisanal academic labs can now create DNA strands stretching into the millions of letters. Given that virus genomes generally top out in the very low hundreds of thousands of letters and bacterial genomes start getting into the millions, it sounds like we're starting to become capable of synthesizing bacterial genomes. Is that correct?
Bacteria and microorganisms, let's say. So E. coli and yeast are probably the two most ambitious projects right now. And most of the assembly is done in vivo. So the little building blocks, as you call them, the short 200 mers, are done by organic chemistry. So it's very far from biology. Then the little assembly is done in vitro. So it's biochemistry. And then once you get them sort of in the 2 to 4 kilobase range, then they're assembled in living cells. And essentially by the kind of recombination we were talking about earlier, which we might call editing, assembly is a form of editing, and it's assembled and then even put into the right place in the E. coli or the yeast genome.
And is the maximum length of assembled strands growing in rapid and compounding ways that parallel the price drops in gene sequencing and synthesis?
Well, maximum strand length is hard to say when you get to assembly in vivo because you're back to editing. So the metric can't be the length of the thing you're changing. It has to be the fraction of base pairs you're changing. And what's interesting is that's largely limited by design. We can't currently design a new chromosome that is more than, say, 4% different from an old chromosome that will still work and do something new, right?
Because our understanding of the metabolic pathways and the underlying dynamics simply aren't rich enough?
That's right. Evolution typically only changes a couple of base pairs at a time per cell generation. And here we are changing 4%. That's pretty amazing compared to evolution. But still, what engineering and evolution have in common is a lot of trial and error.
So cool as it might be to engineer a multi-billion letter mammalian genome from scratch, we couldn't possibly do that design process today. So as a practical matter, we're less limited by strand length than by our understanding of what the genes would do and how they'd interact with one another in the environment. So with that in mind, what do you expect largely or purely synthetic DNA to enable, say, 10 years from now that we're not currently capable of, whether it's for a lack of understanding of the metabolic pathways or an inability to stitch things together in as error-free and efficient of a way as we'd like?
Well, 10 years is a pretty long time in this exponential field that we're in. My prediction is that we won't be purists. I think we will be pragmatists, just as we are today. And if we're given a choice between making 3 billion base pairs of human DNA that's purely synthetic and making a set of 3 billion contiguous bases, we say, well, let's make 3 million changes, but do that in 1,000 different contexts, and they all have an impact. And so now I can make 1,000 different genomes, each of which has been changed quite radically. And then we'll also use that synthetic capability of building test circuits. So we're not just building a thing, we're building all the machinery to test it. And so I think what we're going to be very good at 10 years from now is making lots of prototypes. But these will be more radically engineered because we'll be better at design. So I think we'll be at the point, maybe 10 years from now, where making billions of genomes that are radically altered and still have new functions will be taken for granted.
I'd like to move to a subject that has significant connective tissue with xenotransplantation, literally and figuratively, I guess, which is synthetic meats. And for the benefit of listeners who aren't familiar with this, these are meats that are grown entirely in the lab, no animal involved. So this is quite unlike the pigs growing transplantable organs in that there'd be no conscious entity living, dying, or suffering in the creation of this meat. So, George, what's the status of synthetic meats today, and what breakthroughs do we need to get to lab-grown meats that can both pass a blind taste test and ship at consumer price points?
Well, so it's holding us back is the recipes for making the media to grow the cells, okay? So those recipes typically involve fetal calf serum. So fetal calf serum, just by the name, you can tell that sounds expensive. A fetus, first of all, is a rare, small thing, and getting serum from that fetus,
It sounds like a real process.
and it is. And the replacements for it, where you'll purify the proteins, even more expensive. But there's no particular reason why you can't make cells that don't need that stuff, right? Because all those things are signaling molecules that tell the outside cell, grow, grow, grow. You can just tell the cell directly to grow internally. Or you should be able to make that media for cheap, right? In principle, everything that's made biologically in the world is extraordinarily complex and free. You can go out into the wild, and as far as the eye can see, dozens of kilometers, you see forest and farmland, which is it's benefiting from the sun and the air and the soil, and it's pretty close to free. And by the time it's delivered to the consumer, a lot of biologicals on the order of dollars a kilogram. And that's what everything should be. Everything made biologically should be dollars a kilogram.
But for now, at least, fetal calf serum is a necessary ingredient. It's expensive as hell, and we don't know how to synthesize it.
There are now cell lines that are serum free.
Meat cells that can grow without the expensive serum.
Exactly. That's a big step. And then it becomes a matter of scaling up. And part of the reason I'm excited about it is I've been excited about bringing down prices radically. We've already talked about bringing down sequencing and synthesis by more than 3 million fold. I'd like to see manufacturing of things in general to be brought down to dollars a kilogram. And the more we think about manufacturing as either biological or biologically inspired, the more we can think that's a reasonable goal. And meat is just one thing, but we should be able to make organs for dollars a kilogram, right? Because organ is just meat. It happens to be meat that hasn't been killed yet, but in principle, if you grow it the same way. So I'm very excited about this is the next price revolution. If we can bring down the cost of meat by thousands fold. We're not even asking for 3 million fold this time. We're just asking for thousands fold. We're already down in the couple $1,000s a pound range.
Currently, thousands of dollars per pound for synthetic meat that's what? Good enough for most palates?
It's molecularly identical.
Molecularly identical? Wow.
Vegans already had a solution for this, which is eating vegan burgers, and there's some pretty good ones. There's a transition where a lot of people have their favorite recipes, and it has to taste exactly like beef or chicken or snake or whatever your favorite meat is. Yeah.
Ridiculous as it feels to say only in this context, the fact that you only have to drop down 3 orders of magnitude in price feels pretty doable in this crazy world of synthetic biology. And it probably goes further than that to eventually being way cheaper. And also building on what you said about engineered organs not only being abundant, but resistant to cancer and senescence and pathogens, there's really no reason why synthetic meat couldn't be engineered to be much healthier and tastier than the real thing. And when you combine that with the ethical dimension of no animal suffering, I could see the market for real meat eventually becoming like, I don't know, the market for vinyl records, expensive, artisanal, and retro. And some people swear they can tell the difference, but most people would rather have the thing that's cheaper and better, right?
Right. So almost all the reasons that you want to be vegan could be addressed, including the zoonotic diseases. This is sometimes called clean meat, not just virtuous, non-cruelty meat, but clean, in that when you streak out on a Petri plate, your best meat from your favorite grocery store, it's just covered with bacteria. But this, you streak it out and there's nothing. And that's a plus. You can make it low cholesterol. You can make it so that it's not hurting the environment as much. So beef right now uses up about 20 times as much of the environment as the equivalent vegan resources do.
Remind me which synthetic meat company you're involved in?
Memphis Meats.
And knowing what you know about where they are now and the trajectory of the science in general, do you think we're within 5 to 10 years of synthetic burgers that are as good and as cheap as cow burgers?
I think we're very close. I would be reluctant to say it's a lot more than 5 years because it doesn't require the kind of breakthroughs we need in some of the other things that we've been talking about, like the brain initiative, where we want to be able to read every neuron in the brain. There's some serious technology development there that we can't spell out. When we talk about reversal of aging, we think we have a short list of genes, but working out all the interactions among all those genes and getting good delivery to all the cells in the body, there are going to be some wins before that. But really getting that solved, that's unpredictable.
Wrapping up our wild and woolly topics connected to animals, if you'll forgive the pun, what's the latest with De-extinction and the woolly mammoth project?
We're hoping that this year, 2018, will finally be the year where we publish a lot of peer-reviewed articles that are relevant. And the things that we've got are computational analysis of 23 elephant and mammoth genomes. That's a lot of genomes.
And for folks who aren't familiar with the De-extinction project, you've been able to recover fully intact woolly mammoth DNA.
Yeah.
And woolly mammoths died out 30,000 years ago?
Some of them did. The most recent die-off was around 3,000 or 4,000 years ago on Wrangel Island. But a lot of the genomes we're working with are older than 30,000. That's a revolution all by itself, and that's past tense. That's not wishful thinking. The sequencing technology we talked about earlier has been used to sequence lots of extinct species, including mammoth.
And Tasmanian tigers and passenger pigeons and Neanderthal humans. And in the case of the woolly mammoth genome, you precisely know how it differs from the genomes of existing animals like Asian elephants and African elephants, correct?
That's right. So our project is very pragmatic, as all of our projects are. The goal isn't to necessarily de-extinct a species, but to develop a method by which we can invigorate current species. We're using extinct DNA. So we're essentially de-extincting genes rather than species. And this can help many species other than elephants because a lot of them are endangered because they went through a population bottleneck. They're missing diversity. And even if you go around the world, there's limited diversity. But if you go back in time, there's all kinds of diversity that you can get. And secondly, we can help modern endangered species because the environments are different. For example, for elephants, we'd like to extend their range into the cold because this vast Arctic region, which is underpopulated with herbivores, we depleted it of herbivores that were valuable to the environment.
And what herbivores used to be up there other than the woolly mammoths?
Caribou, elk, bison, small horses, so forth. And they're being repopulated, but they are much less than they used to be, possibly because the keystone species, the mammoth, was responsible for keeping the ratio of grass to trees high. And that has all kinds of consequences because even the biggest non-elephant herbivores can't knock down the trees, and the forests are impenetrable. You can't even fit between the trees. And also, they don't photosynthesize well, so they're not capturing carbon well.
The trees.
And the snow comes in and kind of makes this big, fluffy tree snow blanket. And what happens is in the summer, the temperatures get up close to 20 degrees centigrade. And then the snow and the trees protect it from the minus 40-degree wind in the winter. And so it doesn't cool down well. And we want it to cool down because there's more carbon trapped in the Arctic than in all the rainforests of the world put together, plus all the carbon in the atmosphere.
You mean in the permafrost, right? How much carbon is trapped in there?
The permafrost has 1,400 gigatons, which is like 2.5 times all the tropical rainforests. So letting that melt would be a disaster. What we really want to do is put more carbon in there so the grass can do that. They can keep fixing carbon. So we want to favor the grass, both because we can keep the current permafrost frozen, but also the grass in the summertime will add carbon to that.
So let me play back the sequence just to make sure I'm understanding it. The forest has become impenetrable up there because we don't have these big old mammoths knocking over trees anymore. And the high tree layer insulates the ground, trapping the summer heat, which drastically raises the risk of the permafrost thawing.
Right.
And all these trees crowd out the grass, so we don't have elk and other ruminants devouring grass, which would suck much more CO2 from the air than these trees. So now I see why you want giant tree-wrecking mammals up there, whether it's woolly mammoths or elephants that have some mammoth-like properties that'll let them live much further north. So it sounds like there's a lot of follow-on effects if you can pull this off.
So this would be good for the elephants and good for the environment and good for humans. But the challenge is the closest species to the mammoth is the Asian elephant. The Asian elephant is endangered. We could go to the African elephant. It's close enough. Probably all three of them are interbreedable. I mean, there's some evidence that you can interbreed those. Actually, a lot of evidence that there were hybrids. But even the African elephants, we really don't want to interfere with their normal reproduction. We want African elephants and Asian elephants to be making baby elephants. And so figuring out what genes were selected for in the short distance between elephants and mammoths, it was a very short genetic evolutionary distance. We've got a short list. It's about 44 genetic changes.
That's it?
That's the list that it looks like if you look at their genomes. We can change those easily with CRISPR. We're already doing it in pigs. We can do it in elephant cells. But then we want to grow those into embryos and fetuses without interfering with normal reproduction of elephants. And so we're taking another ambitious project, which is to try to get mammalian development to work entirely in vitro in the lab.
Entirely in the lab, so not with surrogate mothers.
Yes, that's right. That's the goal.
I hadn't realized that. So why not use surrogate mothers, given that you have these closely adjacent species?
Well, they're endangered species, and even cloning in pigs, which is a pretty well-established agricultural cloning species, doesn't have that high a success rate. You know, if you get success rate around 10%, you're doing well. And probably for elephants, it'll be even lower, at least to start. And also pigs, when you get a litter of pigs, I mean, you're talking about, a dozen piglets, and fast too. You're talking about 3 months gestation, and mice is like 20 days. Elephants is 22 months, and they have one child, and then they'll nurse it for a while, and it'll be quite a while before they have their next child. So you can really put a dent into them by lowering their fecundity to 10%.
Yeah, so if there aren't many Asian elephants to begin with, then we use the few that we've got to try to create mammoths, and there's a low hit rate. We could really whack the Asian elephant population. How many are there?
I estimate there's probably on the order of 17,000 that are in prime reproductive years and are female. Now, there's hundreds of thousands of African elephants, but it's still limited. And there's another reason to it is that if we want to scale this, if we want to have 80,000 more or less at once,
Woolly mammoths?
woolly mammoths, or let us say cold resistant elephants that satisfy the woolly mammoth range, if you want 80,000 at once,
That's a lot of 22-month gestation cycles.
yeah, there aren't enough mothers that you have access to, even if all the relevant governments said that this was a good thing. So this is a better way of scaling.
So you'd grow these prenatal critters entirely in the lab and not in a womb. Would you somehow get them to gestate in, I don't know, something like an egg?
We're not necessarily talking about eggs, but it's something like that. It's full development outside of the body with adequate blood supply and nutrients.
So full prenatal development outside of a womb, like completely in vitro. Has any scientific team ever managed to do that for an animal that doesn't come from an egg?
No, there's a lot of reproductive technology. So human preemies now survive from almost about halfway through the gestation. That's coming in from the end. Going forward, there are some mammals that grow essentially free floating. They're not hooked up to an umbilical cord. So you're eating a few days from that end, okay? And we're trying to meet in the middle. So we're working out in mice, which have only 20 days gestation. And again, you can get lots of mouse embryos. And that's pretty easy to do. And we're getting better at turning stem cells into embryo-like structures. And we're getting better at turning embryos into support structures that are vascularized. So we're making vascularized decidua using the same kind of technology we use for making human organ and organoid-like things in the lab. So it's just another organ. It's like making cardiac tissue, we're making brain tissue, we're making decidua, which embryos can implant into. And then once that's working well for mice, we'll try moving into larger animals.
When do you imagine the first woolly mammoth will be born?
Again, we're not aiming for woolly mammoth. We're aiming practically for 80,000 cold elephants. Now, that's ambitious enough.
That's plenty ambitious. When do you think that might be?
The first success could be within a decade. We have to work out all this stuff in mice, then we have to scale it up. And then at a minimum, there's 22 months for the gestation. It's going to take a while. That 22 months is a big problem with getting feedback and doing a cycle of innovation. But that's why we're doing it so much in mice first. So 20 days is a reasonable period of time. Yeah.
So I'd like to close by talking about human longevity. More and more labs are starting to dedicate themselves to combating aging now. So what do you think the landscape of human longevity will look like in 10, 15, 20 years? And what vectors are we pursuing right now that could have a real impact on how long people live?
There's two major strategies. One is aiming at extending longevity, and the other is aging reversal. The problem with longevity extension is if you're not careful, you extend some of the weaker years of your life. Nobody really wants that. Aging reversal, on the other hand, sounds a little more speculative, but there are several cases of it in mice. You can get aging reversal with blood-borne components, essentially a kind of a transfusion from a young to an old. You can get it from the ends of the chromosomes, telomeres. And then there's a way that you can return old adult cells to embryonic stage. And if you do that in limited doses in an intact mouse, you get some evidence of aging reversal. So those are three examples. And then there's a vast literature on longevity as well in many model organisms, flies and worms and mice. And so we're taking that literature and we're asking which of those could we turn into gene therapies to try aging reversal in a gene therapy form in mice. You can summarize the different ways of aging in about 9 different categories, and you might have to fix all 9 of those things simultaneously.
That sounds a lot like Aubrey de Grey's approach.
Well, his is more focused on damage, and mine is more focused on epigenetics. I'm sure there's damage, but the fact is that young cells respond to damage better, fix damage better. And so I'm just trying to convince old cells they're young cells and then let them handle the damage.
In other words, if an old cell becomes young, it's innately configured to fight damage.
So Aubrey is a very complementary approach. I'm one of his advisors to his group, but it's a complementary thing. Is if we need additional damage control, they will do it. And so we're basing it on all the examples of either longevity or aging reversal. We're reformulating it as aging reversal gene therapy. turning gene therapy into an aging reversal drug. And so we've got about 40 now in the pipeline. We have tested on pre-aged mice. These are aging reversal gene therapies that have been chosen to be compatible with where we are right now for gene therapy, which is we can't treat every cell in the body. So we have to pick the subset of genes where we don't have to treat every cell in the body. And that's what we want right now, because most people that are worried about aging are not the unborn.
Right.
And so our market are people that are old. And so we're doing it on pre-aged mice that are like 2 years old, almost dead. The ones that work there, we're moving into dogs, pre-aged 10 to 12 years. And that's a market in and of itself. That's not only a good test bed to move up from mice to something that's more like humans. Dogs live in human environments. They get human attention. It's a nice pause point for the technology because we can get feedback from pet owners. They will actually do our scale-up preclinical animal trials for us, and they'll pay for it. So it's a win-win all the way around. That's the business model for the company that we're starting called Rejuvenate Bio. But we've already done a bunch of trials in mice, and we're doing some in dogs. And then we'll move on to humans.
And the company's fully up and running?
Well, it's up and running in the sense that we're doing all these experiments.
Have you seen anything eye-popping from the experiments yet?
We're publishing a paper where we did 3 pretty eye-popping experiments in various combinations.
And how long do you imagine it'll be before you start seeing major results in dogs?
I have to apologize for everything seems 5 years away, because that's almost a cliche.
Hey, it's fine with me. I want you to be right about all this stuff.
But I'll tell you why I'm less embarrassed than I would have been. I've seen a bunch of things that even the optimists felt were 6 decades away, like affordable sequencing. When it was $3 billion, 6 decades was pretty optimistic. It was assuming that we could get genomics on a Moore's Law curve and then keep it on the curve for 6 decades. And instead, it took 6 years. And so when I say 5 years, I have a few examples in my pocket that are past tense. Sequencing and synthesis and CRISPR was even less than 5 years. The clinical trials for dogs is much shorter. And we're talking about aging reversal. That's another reason to do aging reversal rather than longevity, is that it's hard to get FDA approval, even for a veterinary drug, because if you say it's going to extend the dog's life by 10 years, that's a 10-year clinical trial, which if you say in 5 weeks it's going to make them stronger and more resistant to injury, then that's a 5-week.